Hello friends, warm welcome to all to our channel World of Zoology. This is our first lecture on the new chapter Instrumentation, and the today's topic of interest is Introduction for the topic Instrumentation. Objectives: to give learners an insight about biological structures and train them with operational skills for using different instruments required in zoological studies. Desired outcome: the students will be skilled to select and operate suitable instrument for the studies of the different components of zoology of this course. Biological processes depends upon various types of chemical reactions such as acid base or neutralization reaction, oxidation reduction reactions, that is redox or electron transfer, the various definitions of which are as you know that, and hydration, rehydration, dehydration, the reactions involving properties of the solvent water. Survival, sustenance, and even the replication of the life forms needs a fine control over these reactions. Therefore, fate of the reactant and their products need to be carefully analyzed in order to understand a given biological process. The importance of the concentration of chemical species in biological events could be seen in a way the human body controls hydrogen ion concentration in blood via different buffering systems. The measure of concentration of hydrogen ions, that is expressed in terms of number of pH units, gives us some idea of such control. At normal body temperature, that is 37 degrees Celsius, the pH of the blood ranges from 7.35 to 7.45, that is 10 raised to minus 7.35 to 10 raised to minus 7.45 moles per dm cube of hydrogen ions. A variation of as much as zero point 0.3 from these normal values could be fatal. The example which is given of the blood pH. When pH goes below 7.3, acidosis sets in, during which the central nervous system starts to malfunction and individual becomes disoriented, and even that leads to comatose condition. When the pH goes about 7.45, alkalosis sets or results in a major effect of which is hyperexcitability of nervous system leading to spasm and tetanic contraction, that is bending of the muscles. Severe alkalosis can cause death due to tetany, that is intermittent tonic contraction, and severe muscular pains due to abnormal calcium metabolism of the respiratory system. It is due to the convenience of expressing the ionic concentration in terms of numbers 7.35 to 7.45 instead of moles per dm cube that help us to understand such biological consequences. We shall learn more about the concept of the pH as a unit to express concentration shortly. Biological systems have several mechanisms to control chemical reactions. Because billions of reactions which are taking place in our body, we are unaware of the fact the particular pH which is required for the particular enzymatic reaction is different from the other enzymatic reactions. So, say for example, in case of the stomach, the pH is 3, that is acidic, whereas the within the buccal cavity, the biochemical reaction or the enzymatic reaction that occurs, such as action of the enzyme salivary amylase that takes place at pH 6.7, that is more near to about neutral pH. The product of such reactions that are useful have to be retained, and those products which are useless or harmful to the body have to be eliminated. Complex molecular systems such as enzymes control a specific chemical reaction. For example, enzyme maltase catalyzes the conversion of maltose to glucose. Our knowledge about various biological systems and hence of the life science is enhanced by learning the mechanism of the various chemical reactions occurring in the living system, what we term it as the biochemical reaction, and the origin and fate of the chemical species that are either involved or are formed in such reactions. Therefore, chemical analysis of the biological systems becomes very important and indispensable. The science of chemical analysis has come a way long from using human senses alone to learn about the type of such chemical species. Our five sensory organs, tongue, then skin can judge the temperature, even the eyes, nose, and tongue. These are some of the sensory organs, including nose. Here, tongue, for example, give us an idea about the hydrogen ion concentration. Higher the hydrogen ion concentration means that particular substance is acidic. Acids are sour and pungent. Lower concentration of the hydrogen ions means substance is basic. Bases are bitter and could be soapy. The taste buds on our tongue are thus able to differentiate substances as acids and bases. Similarly, the human eye may serve as detector for estimation of the color intensity of a particular sample, the amount of analyte that may be present in the sample. An analyte is a specific component in the given sample being studied. Development of sophisticated instruments has come a long way in facilitating various types of investigations of the biological samples. Broadly, a biological sample can be studied so as to observe and understand its fine structure. Secondly, to isolate or separate from a mixture of the another molecules or compounds. Separation techniques such as filtration, centrifugation, then chromatography, electrophoresis, that varies according to the need. Analyze for the third one, that is analyze for the amount of the certain component with the help of the various instruments such as colorimeter. Here the instrument which is used for this purpose may be represented by the diagram shown below, in which a diagram of the analytical instrument is given, where the stimulus for the source such as a light, heat, current or voltage is shown that passes through the sample, and then input which is detected with the help of the detector. Then amplification of that particular signal is carried out with the help of the signal processor or digitalizer. Then the particular result is read out with the help of the meter, plotter or the computer, which is shown here. Fourth one, that is the particular biological sample which is studied so as to understand the molecular structure of a given molecule of biological interest. That particular molecule may be protein, again enzymes which are protein in nature, or the hormone or the DNA, with the help of the spectroscopy such as infrared, nuclear magnetic resonance, electron spin resonance, X-ray diffraction, etc. To understand a given process as it occurs in biological system with the help of the kinetic studies. Thanks for watching this introductory lecture on the topic instrumentation. Do like if you like the lecture. Share it for the benefit of the maximum student and subscribe the channel World of Zoology for the notification of the next lecture on the same topic, instrumentation.